The overall goal of this procedure is to study mammalian cell division and cell matrix interactions in a physiologically relevant 3D environment.
The main advantage of this technique is that it provides an efficient and general approach to study mammalian cell division and cell matrix interactions. This method can help answer key questions in cell biology, such as the molecular basis of the development of normal and diseased tissue.
A day prior to transfection, plate 5 million HEK293 T cells in a 100 mm cell culture dish in 10 milliliters of normal cell culture medium. Incubate these cells at 37 degrees Celsius for 24 hours in a humidified incubator with 5% carbon dioxide. The next day, just before starting transfections, remove a vial of the transfection reagent from 4 degrees Celsius and leave it out to reach room temperature. Then, to start, add 1 milliliter of reduced serum medium to a sterile microfuge tube. To this, add specified amounts of lentiviral vector plasmids used for transfection and mix by pipetting. Incubate the tube at room temperature for 5 minutes. After 5 minutes, add 48 microliters of the transfection reagent into this tube and mix gently by pipetting. Subsequently, incubate this mixture at room temperature for at least 15 minutes. Once the incubation is complete, remove the plate with HEK293 T cells from the incubator. Add the prepared DNA transfection reagent mixture dropwise onto the cells and gently swirl the plate to ensure even distribution in the plate. Place the plate back into the incubator.